So thanks to all of you for coming this morning to the Knight Alzheimer's Disease Research Center Participants Breakfast. We're delighted to see all of you. We look forward to this every year to give you an update on the progress we have made in our research. And all of that progress that we've made depends upon all of you. So we want you to take ownership of what we're doing, because without your participation, we would not be able to have any progress whatsoever. So we thank you and appreciate all of you being here. Now, a couple of housekeeping notes. First, I think there's even a slide to verify this. Will you turn off or put your cell phone on vibrate? Second, you probably recall, for those of you who attended this breakfast before, that after our speakers all have completed their presentations, we'll have them all join me up here on the podium, and we'll open it up to you for your questions. And at your table should be note cards and pens, and we're going to ask that you write your questions down on the note card or index card, and our staff will come by and pick it up and bring your questions up here, and we'll have the panelists then address the questions. So, we have a terrific group of participants, you. We also have a terrific staff at our Memory and Aging Project, and many of the staff are here today and are very much involved in ensuring that this participants breakfast is a success. They put a lot of work into it. You may have seen people out in the rain and drizzle to be sure that you found your way here appropriately. And I'd like all the Memory and Aging Project staff to stand, including our faculty. Please stand, everyone. Laura Swisher is in from the rain now, is that right? She just made it in. She was at the far outpost. Now, so all of the staff and the faculty have worked hard to put this breakfast on, but one person has to be in charge of it and take charge of it and be responsible for it, and that's Angela Oliver, and she's in the back standing, waving her hand high. Yep. So thanks to Angela. Now, I mentioned writing your questions on the index card. Some of you who have attended this breakfast on a regular basis may remember a question that came up when we talked about home safety. One of our presenters, Danielle, presented on home safety and showed, if you remember, a slide of someone living alone in her apartment and trying to get up on a step ladder that looked pretty shaky to change the batteries in her smoke detector. And that, of course, is a real danger for terrible fall. So one of our participants took that to heart and asked me to share with you that she found out that her local fire department, if you call them, would be happy to come to your home if you supply the batteries and change the batteries for you. So I thought that was terrific, and so don't get up on that step ladder. Call the fire department. All righty. Let's set the stage for what we're going to hear today. So our research is almost 100% funded by a part of the National Institutes of Health. It's called the National Institute on Aging. And it is supporting all of our research studies. It does so by awarding us funds through a grant mechanism. Now, this is competitive. We apply for the funding, and we're judged by independent reviewers, as to whether our application for funding is meritorious. We are fortunate that we have 4 large grants presently from the National Institute on Aging. The first is the actual Alzheimer's Disease Research Center grant. That was first funded in 1985, and has been continuously funded since. Matter of fact, we just began this month year 31 of continuous funding of the Alzheimer's Disease Research Grant. Now, you can see that in 2010, we were very fortunate to have Charles and Joanne Knight endow the center to ensure its financial stability. And so now we call it the Knight Alzheimer's Disease Research Center. And Joanne Knight is with us this morning at the front table, so let's thank her and recognize her. Another large grant is called Healthy Aging and Senile Dementia, and that began in 1984 and has been continuously funded. The Adult Children Study began in 2005, and the Dominantly Inherited Alzheimer Network began in 2008, and all have been successfully renewed. We have to reapply for this grant money every 5 years for each of those grants. It's a lot of work. Now, you probably, when you come to participate, don't know anything about the grants, because no matter which grant is funding your particular research, everyone comes to the central research office called the Memory and Aging Project. You've heard of that, or MAP. So MAP supports all of these grants by providing the participant evaluations. Now, here's the faculty and staff of the Knight Alzheimer Disease Research Center. Not that I'm biased, but the top Alzheimer Disease Research Center in the country. There are 28 of us, and I really think our faculty and our staff make us number one. We have some 80 faculty who have part of their work supported by the Alzheimer's Disease Research Center, and maybe 40 staff who are fully supported by the Alzheimer's Disease Research Center. And with that large number of people come and people leave. So we welcome 2 physicians to our team, Dr. Lenice Cummings-Vaughn, who is on maternity leave, so is not with us today, and Dr. Greg Day. Did I introduce Dr. Day last year? Is Dr. Day here? Do we know? Yep, there's Dr. Day. We also welcome new staff: Tiffany Earle, Annalise Hernandez, Jenny Petros, Liz Rourke, Laura Swisher, and Sarah Stout. And we have visiting us for a year a neurologist from Tokyo, Dr. Ryoko Ihara. Ryoko, you want to stand, please? So she will be training with us to learn our assessment techniques, as well as our approach and our administration of this research program. I said going is Dr. David Carr. Now, many of you know Dr. Carr. He is not leaving Washington University, but he has taken on increasing leadership responsibilities at the St. Louis Rehabilitation Institute. So he is leaving the Memory and Aging Project, but still will be at Washington University. And then 3 of our psychometricians, the people who administer the memory and thinking tests, are leaving: Rachel Chassie, David Rivolo, and Kat Vernon. But Rachel really is leaving the Memory and Aging Project, but not the Alzheimer's Disease Research Center. She's joining another part of the ADRC, and Kat Vernon is still with us as well. So I think Rachel and Kat are with us. Are they? Will they stand if they're with us? There's Rachel and... All right. Now, what's our theme? We have many different research projects. We address many different questions. But overall, for several decades now, we've been pursuing the idea that Alzheimer's disease is a chronic illness, a long illness, maybe 30 years in duration. But much of that duration, the brain changes of Alzheimer's disease occur but don't produce any symptoms. So it's building up in the brain, but not enough brain damage has occurred to cause the memory loss and other thinking changes. So there's a silent phase of Alzheimer's disease that we cannot detect because there's no memory loss. So we call this preclinical Alzheimer's disease. It's not yet clinically expressed, no symptoms. For the past 15 or so years, we've been employing markers of the brain changes of Alzheimer's disease in people who have no symptoms, and we can identify that the brain changes are present. The two major biomarkers, that's what we call them because they detect the biology of the pathobiology of Alzheimer's disease in the brain in the absence of symptoms. The two main ways we do this are sophisticated imaging, including PET scans, and analysis of the cerebral spinal fluid, because the spinal fluid has the proteins in it that represent those brain lesions of Alzheimer's disease. So we can find out whether people have the brain changes of Alzheimer's disease before symptoms. Our goal is to determine, will everyone who carries those brain changes, if they continue to live, will they develop the symptoms of Alzheimer's disease? Is that a 100% process or not? Can we characterize this period of time of silent Alzheimer's disease and identify people who are on the road to developing symptoms? Can we identify those individuals? We think that's very important because we would like to initiate therapies in this silent phase in the hopes of delaying the onset of symptoms or even preventing that, preventing Alzheimer's disease symptoms from occurring. Prevention studies. You know that we've had a tough time in the field of trying to find truly effective medications once the symptoms have declared themselves. We haven't been very successful in doing that. And it may be that by the time symptoms occur, there's so much brain damage that medications may not be effective. If we can go earlier in the process, maybe this medications will be effective because the brain damage has not yet fully expressed itself. So prevention trials. We launched, under the direction of Randy Bateman, the first ever prevention trial in people who are without any symptoms whatsoever, but are at very high risk of becoming symptomatic if they continue to live. That started in 2012. And Dr. Joy Snyder is going to talk about another prevention study that we've launched as part of a large group. We're one of many sites that are participating in this national study, and it's called the A4 study, and you'll hear more about that. Dr. Bateman is our site principal investigator for the A4 study as well. So let me tell you a little bit about you. If we add up all of our participants in all of our grants, we have over 900 participants. Now, people who are 65 and older enroll in the Healthy Aging and Senile Dementia or the Alzheimer's Disease Research Center grant, the first indentation there, 538 people in those two grants. And look, almost 350 are cognitively normal, and about 200 have Alzheimer's dementia. This reflects our emphasis on identifying the disease in the clinically silent stage. So we're an Alzheimer's center, we're still investigating Alzheimer's disease, but the emphasis, as you can see, is on people who have yet to develop the disease. We want to try to identify those using biomarkers who are at very high risk and try to characterize them. The adult children study is about 300, and our Diane, the dominantly inherited, is about 83 at Washington University. It's an international study, and there are about 420 international. So you can see we're by far the largest enrolling site in Diane. Look at that. The age range is 22 to 101. We're really studying the disease throughout the lifespan. It's amazing, because we really want to pick up when those first brain changes seem to occur. About 57% women, 18% African American. I want to once again acknowledge the fact that we strive to represent the St. Louis metropolitan area in terms of its characteristics, so that we represent the entire population. If we only study Alzheimer's disease or preclinical Alzheimer's disease in Caucasians, we won't know whether that translates to African Americans or other underserved minorities. And we had a real challenge when we began our diversity program to overcome some barriers to African American participation. So we convened an African American advisory board, and they have been remarkable in assisting us in helping us be more welcoming to people of color. The original chair of the African American Advisory Board is Mr. Norman Say, and he is seated here at the front table as well. And there are several members of our advisory board in the audience. Mr. Sidney White, Miss Sally Simpkins. My memory isn't so good, so if I've missed some people, I apologize. 3 people have come to our program for 30 years in a row. Isn't that remarkable? And people come from all over. Oklahoma, Idaho, Maine, California, Texas. So it's a remarkably you are a remarkably dedicated group, which we very much appreciate. So what do we ask of you? Well, I'm going to be blunt. We ask an awful lot. Matter of fact, I don't know any other Alzheimer's center that asks so much from you. And you provide it to us. It helps us immensely in our research, which makes us the top Alzheimer's center. So what we ask is every year that you come for an evaluation, and every 3 years that you undergo the imaging and the spinal fluid assessments. I want to emphasize that coming back is essential for our research. Essential. We need to know what the memory status is for each of our persons so we can make our biomarker information understandable. Erica Keys is our recruiter. By that I mean she's the person that calls people to see if when they can be scheduled to come back. Erica is standing up. She's waving her hand. She is terrific. We understand, by the way, that you have a life outside of the Memory and Aging Project. And people take vacations, go visit grandchildren, sometimes they're in the hospital. But when Erica calls and leaves a message for you, please let her know, call her back, let her know your circumstances. Even if you can't come and be scheduled now, please let her know so that she can make a note and call you at a better time. If you move and have new contact information, please let us know that as well. We can't keep up with you unless we know where you are. Now, something that's been a little worrisome to me is once people are scheduled, they call to cancel, and again, we understand that circumstances don't always permit people to keep their appointment, but the cancellation rate has been increasing from about 14% to 21% over the past 4 years. This has major influence on our ability to continue the research because it is the accrual, the ongoing data collection that allows us to do our job. So here's the participation rates of some of our biomarkers. A brain MRI scan, when you first are enrolled, almost everyone gets that completed. The PET scan to look for these brain lesions in the clinically silent phase, about 2/3 complete that. And look at the initial lumbar puncture, 80% complete that. That's remarkable. You are terrific. But look at the follow-up rates. They fall quite dramatically. And I just wanted to, and there's also, we're just starting to enroll for a sleep study that began last fall. Dr. Brendan Lucy is here, and he is recruiting for that sleep study. So I'm not going to trivialize this. All of these studies take time on your part, and they're not always easy to do. I can appreciate that. But let me illustrate why it's important for the research. So this is the PET imaging data for the brain amyloid. Brain amyloid. And this red line going across is the determination between normal versus abnormal brain amyloid. You know, what we hope to have is no brain amyloid, 0. But what we're seeing are some people are accumulating amyloid in the brain. That's what these upgoing data points show. And we think that brain amyloid is a key lesion that will cause individuals ultimately to develop symptomatic Alzheimer's disease. Now, what we're showing here, Dr. Andrei Vlasenko is here today as well and did these analyses, are brain PET scans in people who had 2 PET scans, about 3 years apart. Now, that's a lot of people had at least 2 PET scans, but it, believe it or not, may not be enough. So let's take a look at one person right there and say they had a 3rd PET scan. It looked originally like they were going down, but the 3rd PET scan would show us that really they're about the same. They're holding steady. We wouldn't know that without the 3rd PET scan. Let's take another person. Let's take this person. Looks like they're going down in their brain amyloid level. Well, maybe it's plateauing. Without a 3rd PET scan, we can't tell that. And yet here's another person. Is it possible if you have amyloid in the brain that it can get cleared away? It can go away? Well, we're only going to know that if we get multiple PET scans. So 2 are great, 3, 4, much more informative. All right, so I said we ask an awful lot of you. we recently took a poll, and we're going to do another survey, by the way, this summer, that we want all of you to participate in and tell us how we can be less demanding of you and your time. But I was surprised that it's the memory and thinking tests that our people, our participants, dislike most. They don't like it. You don't like it. They'd rather have a spinal tap. Now, we've given a lot of thought to this, and I can appreciate, particularly if you're concerned about your memory, or maybe had a relative, a parent who had memory problems, that at some point during that testing, you're not going to do well. You're not. And that could be alarming. It could say to you, oh my gosh, well, I should have remembered that. They gave me a list of 10 words and I only remembered 7 or something like that. And it can be upsetting.
That's a lot.
But here's what I want you to know. These tests are designed for you to fail. Everyone's going to fail. You should not be concerned that if you remembered 6 out of 10 or 7 out of 10 or 3 out of 10. That's what they're designed for. We're trying to find out how people's memory goes. No one is expected to get everything right. Don't worry. Now, when I showed these results to our psychometric testers, they got pretty upset. So please don't shy away from the memory and thinking test and make our psychometricians happy again. By the way, they're a terrific group of individuals. They're led by Denise Maui-Dreyfus and Jason Hassenstab, and all of our testers, I think you will agree, are very supportive. Here's another reason I'm a little concerned about the increase in cancellation rate and the lower percentage of follow-up completion of our studies. There are a lot, because you're such a great group of participants, so dedicated, so committed, and have such rich data from the biomarkers, lots of other studies like to utilize you in their studies. So we have at least 11 additional studies outside of our own memory and aging project that are calling to see if you want to participate. A driving study, a hearing study, a memory study about things we have to do in the future. And here's some of your comments. Which study is this for? I've already been contacted a lot. I've got a lot of study dates on my calendar. Boy, there's a lot of research right now. So as I mentioned, our education corps, led by Andre Denny, is going to ask you for your opinion. Maybe we have asked too many studies. And you've heard me say, and this continues, we know that some of you would be motivated to participate in research even more if you had access to your own research results. And we're very aware of it. So we have not, there are lots of, I think I've mentioned in other settings, there are lots of considerations about that, but please do this study because we want to have your input on that as well about getting your own research findings back. Okay, so we've been very fortunate, as I mentioned, through the National Institute on Aging to get this funding. Matter of fact, right now I am the #1 best funded individual from the NIA throughout all of neurology. But not just me, Dr. Randy Bateman and Dr. David Holtzman are also in the top 25 in the entire field. And it's amazing. I don't know how they do it. Dr. Bateman is still quite young. He's in his 40s, and he's already a top researcher. David Holtzman's the chair of the Department of Neurology and still has time to do this cutting-edge research, so they're remarkable. So I told you each grant has to be renewed every 5 years. Because our grants are large, we must request approval to put in our application at the budget size that we need to do our research. And the field has changed. I told you that it's a competitive review. A special panel of experts judges whether our grant is meritorious or not. And they think in general that too much money goes to large grants. That's a bias. They look at a senior investigator, that's a polite way of saying me, of having so much funding when other investigators have a hard time getting it. So there's a trend to doing single project grants, not large grants like we have, and an emphasis on early stage investigators. So that the attitude of the funding agencies are sort of stacked against us. Matter of fact, at this point, it's almost a case, matter of fact, it is a case, we can't have any criticisms of our application. We need a perfect score to guarantee that our grant is renewed. Now, here's what grant reviewers look like. These are comments. You know, whether we agree with the critiques that they provide to us or not, it doesn't matter. It's their decision that determines whether we get funded. They say that our clinical assessment follow-up needs to be bolstered. Remember that 21% cancellation rate. We need to bolster that number. Poor rate of follow-up among people who have symptomatic Alzheimer's disease. We're below our completion of follow-up visits compared to others. So these things reduce our score and jeopardize whether we get funding. So we appreciate that we ask very much from you, but your participation going forward is critical to help us advance the field of Alzheimer's research. Now, this may be you may not know this. Your participation has led us to lead the field. The first ever report of preclinical Alzheimer's disease in living people came from us, 2006, Mark Minton. The first report that analysis of that spinal fluid can predict future development of clinical Alzheimer's disease came from us, Ann Fagan, 2007. And the first report that amyloid imaging can predict development of symptomatic Alzheimer's disease came from us as well. And I've already mentioned that we launched the first ever prevention trial in clinically normal people in 2012. This slide comes from Dr. Holtzman. I'll just show you, this is the first ever Alzheimer patient. Her name was Auguste Dieter. These are the original photomicrographs of her brain autopsy showing amyloid plaques and neurofibrillary tangles. The tangles we know is a protein called tau. The plaques come from amyloid beta. This is looking at the plaques and tangles in more modern techniques. By the way, these original photomicrographs came from Alwas Alzheimer. He studied the brain of this lady, and that's why we call it by his name. But what these are the signs of Alzheimer's disease in the brain, but what they end up reflecting is a loss of brain cells. And because brain cells are lost, the brain shrinks. You can see the brain of someone who died of Alzheimer's compared with the brain of someone who didn't die of Alzheimer's. Now they died. It says healthy brain, but I guess they're dead. So, but they didn't have Alzheimer's. And you can see the shrinkage. We call this atrophy, and it means brain cells are gone. So that's what happens in Alzheimer's disease. We can detect these brain changes during life with PET scans. I've already mentioned the amyloid plaques and the amyloid beta protein, and now the neurofibrillary tangles and tau protein. We have tracers that PET scans can detect. We've been using a tracer called Pittsburgh Compound B, but we're moving towards another one called Florbetapir. They both detect amyloid plaque in the brain. And for the past year or so, we've been looking at a tau PET tracer to look at the tau neurofibrillary tangles in people. Now, how many of you have heard that you cannot know for certain whether somebody had Alzheimer's disease until they die and the brain is examined by a neuropathologist? Anyone hear that? No longer. We can detect it in living people. You don't necessarily have to have an autopsy. Now, I've mentioned this already, so I'll be brief. We think there's a long stage of Alzheimer's disease prior to the appearance of symptoms. Matter of fact, the symptomatic stage lasts about 7 to 10 years. That's the end stage of the illness. So we're interested in this preclinical stage, and how do we identify it? Biomarkers, the cerebral spinal fluid, CSF, amyloid beta, amyloid imaging, CSF tau, spinal fluid tau. Now what we think is, so we can detect it, and what we think is they build up and eventually they cause nerve cells to die. Nerve cells are called neurons. When nerve cells die, you're on your way to developing symptomatic Alzheimer's disease. This is a new technique for us. I mentioned tau PET imaging. Dr. Tammy Benzinger, who heads our imaging core, is with us. And we can see that in looking at a diagram, this person had no tau, this person had limited tau, this person had more tau, this person has widespread tau. And this is the tau imaging, and you can see it sort of parallels that. A little bit of tau in the yellow seen here, a little more yellow seen here, and now widespread here. So we can see this in living people, and this is just another way of showing the same thing. Up on top are brain slices of people who died with low, medium, and high amount of tau, and you can see the black now in the panels below, D, E, and F. It increases with it, so it reflects tau pathology. So we need you to help us identify people to participate in our research. Each year we try to enroll 38 cognitively normal people. We're 5 months into this year and we only have 12, so we need more cognitively normal people. We want people who have mild symptomatic Alzheimer's disease. We want 27, we only have 10. And as I mentioned, we want African Americans. And we want 20 or more every year, and we're lagging this year, only 4. So we need your help to encourage your friends, your relatives, to please participate. In general, we want people to be 65 years and older, stable health. Doesn't mean you can't have health conditions, but as long as they're stable. Participate annually, have a study partner, and be willing and able to do our procedures, including the spinal tap, lumbar puncture, and the brain imaging. And there's the phone number to call. But we do ask you to please help us get our sample sizes to where they need to be. And this looks rather stern, so I want you to keep watching this a little bit. So we want you to come back for all of our assessments and our memory testing. Thank you very much. Now at the end of the program, some of the studies that we have that require some extra assessments and extra visits are here today. You're going to hear about the A4 trial. Dr. Snyder's going to talk about that. As I mentioned, our site principal investigator is Randy Bateman. So there's going to be a table out in the foyer for more information about that. I mentioned Dr. Lucy is with us today. He has a sleep study, and he's with us today as well, and he's going to have a table out in the foyer for more information after this meeting ends. Some of you have heard that some of you have participated in a study to see if an eye examination can detect some of the brain changes of Alzheimer's disease. You may not know that the eye, and particularly the optic nerve, is an extension of the brain. So looking at the eye may tell us whether or not there's a signal there. And Dr. Greg Van Stavern is here, and he's going to have a table in the foyer as well. And then Dr. Bateman has a special study of looking at the metabolism of these brain proteins, including amyloid beta. And he is going to be in the foyer as well with another table. So with that, I'm going to now turn to our program. You're going to enjoy this. We have 4 speakers, and the first is Dr. Krista Mulder, and she's going to be talking about the importance of the spinal fluid in Alzheimer's research with a particular emphasis on the spinal tap. So where is Dr. Mulder? There she is. So let's welcome her.
Good morning.
Good morning.
So I just wanted to emphasize from the very beginning, it's hard following Dr. Morris, so I hope you'll cut me some slack. I first wanted to emphasize that we have a really unique, because of you, a really unique study here, that we lead the way in spinal fluid collection among other famous Alzheimer's studies across the country. So I've put the Knight ADRC up here. Oops. Among some other studies, some of which you may have heard of or some of which you haven't, that are international. For example, on the top left here, this is ABLE study. This is actually a study in Australia looking at aging and Alzheimer's disease. And the 2 studies on the left, they're very large studies, but they don't really have a focus on spinal fluid collection. There's some other studies on the right. For example, on the top here, I have Johns Hopkins University, which you may have heard of. They have a study called BioCard. But that had a lapse, and it doesn't have nearly the amount of spinal fluid collected that is from UVAR participants. And there's another study down here from the Wisconsin Alzheimer's Disease Research Center. They have a study called WRAP, the Wisconsin Registry for Alzheimer's Prevention, and they have some spinal fluid. But really, through efforts from Dr. Holzman, Dr. Fagan, and many groups, we actually have the largest amount of spinal fluid. We think we don't have the exact vials counted, but more than any other Alzheimer's study in the world. So I'm going to emphasize some points that Dr. Morris brought up, and we're driving this home a little bit, but we have to think about, you say, well, I did one spinal tap, why are you asking me to do another one? So you may have heard from some other people who have spoken here before that in the spinal fluid, in the preclinical stages of Alzheimer's disease, the amount of amyloid protein actually goes down. We think that's because the amyloid is getting stuck into those amyloid plaques that are in the brain, and that's why you don't have as much of the amyloid protein in your spinal fluid. So this is on the, whoops, using the wrong pointer here. So you can see this is the amount of amyloid, just an example that would be in your spinal fluid. And from year 1 to year 2, it looks like it's going down. So is this person losing the amount of amyloid in their spinal fluid? Well, with only 2 points, as Dr. Morris was emphasizing, we don't know the full story. Whoops. It could be that if you looked in year 3, it really is going down, that person may be developing Alzheimer's disease. Or it could be that it's stabilized, that was just a small decrease, there's not really any change. Or it could be it's gone back up. That was just a fluctuation for some reason in year 2, they had a little bit less, but pretty much they're staying the same. So that's why we want you not just to come back 2 times, 3 times, 4 times. The more time points we can see the true picture of what's happening over time, as opposed to even just 2 points doesn't tell us the full story. And one point that we make is, so of all of the MAP participants, we have only 120 people with 3 or more lumbar punctures, and that's absolutely amazing. 120 is amazing, but we really need it to be, the full spectrum of everyone in the study to have the most data possible.
Are any of them here?
I don't know, have anyone here had 3 or more LPs and want to stand? Please stand.
And notice they are still standing and walking.
And And so we certainly want everyone to come back for their longitudinal visits, the clinical, the psychometrics, all of your biomarkers. But I'm focusing on lumbar puncture here, and you don't have to appreciate everything that's in this slide. But this is from some data. This is actually from our Diane study, but it's looking at what are the things that change earliest when you're looking at what's going to happen for people who will ultimately develop the memory and thinking problems with Alzheimer's disease. One of the things you can see that I just want to point out here is one of the very earliest changes that we can see is this change in that amyloid protein in your spinal fluid. So there are many other things that change before people develop symptoms, but we really are focused, at least for my talk right now, on the spinal tap, because you can see that that's one of the very earliest changes. So it gives us the earliest sign as far as when people might ultimately develop Alzheimer's disease. So that's one of the reasons we're focusing on it. So some of you may have participated in a survey that we did that Dr. Morris helped to lead that took place at Washington University, but at all of what at the time were the 27 Alzheimer's centers in the country. So we wanted to ask, what influences the ability for people to agree to undergo lumbar puncture? So we don't have 100% agreement, so why are people perhaps saying no? Is it the perceptions of the people at the site? If you have a doctor that says, well, I don't think that's very important, you don't need to do that. Is it your past experience? You had a lumbar puncture headache that makes you think, I don't want to do another one? Or are there other things? And then secondarily, I lose my little red dot. What factors contribute to lumbar puncture complications? So we certainly want everyone's lumbar puncture to be completely seamless and perfectly fine, but we know that there are complications sometimes, and what can we do to minimize those? So we conducted a 2-year study at all of the Alzheimer's centers across the country. It was led by Dr. Morris and another individual called Dr. Bud Kukul, who's at the University of Washington in Seattle. So WashU and University of Washington working together. And we had some funding from the National Alzheimer's Association that helped us conduct this survey. So what influences people's agreement to undergo LP? Well, we know, and we're sensitive here in our studies, that race does matter. This is something that's actually very different across different cultures. And as I said, so African Americans and Asians were less likely to agree to lumbar puncture than Caucasians. So that tells us that there are different factors, people have different influences in their lives, that we need to be sensitive to that and have education that's tailored to different people's concerns and needs. We also know that experience matters, and we were actually quite impressed with this. It's actually people who have already undergone lumbar puncture were more likely to agree to lumbar puncture, a second one, than people who had never had lumbar puncture again. So 90% of people who had previously had a lumbar puncture agreed to a second, whereas 73% of people who had never had a lumbar puncture agreed to their first. So we think that tells that if you've had a lumbar puncture, and I have too, I can say that Dr. Snyder, who's speaking a little bit later, did mine. I had absolutely no problems. And so I would absolutely do it again. I know that's not quite as scary as sometimes, if you don't have any knowledge of that you might think that it is. So we think if we just educate people, that there's a greater chance that they'll agree. And also, surprisingly, we found that if someone had had a lumbar puncture headache, that did not change their agreement to undergo further lumbar puncture again. They were the same, agreeing to the lumbar puncture at the same rate as someone who had experienced no complications. So I think that speaks to the dedication of all of our participants. But when does complications happen? So we found that across all the Alzheimer's centers, about 11% of people experienced a lumbar puncture headache. Now I want to say that headaches were generally mild and treated with some caffeine, so some soda or coffee, or oral analgesics, so over-the-counter medication, or just taking fluids to help rebuild the fluid volume. You can have what's called a blood patch, which we can talk about more if necessary later, but that's something that can happen with a very severe lumbar puncture headache, but that was in only 1% of persons, so it's actually a very, very rare occurrence. Now, some people do experience other mild complications, for example, nausea or dizziness. You might have gotten a little lightheaded, and that happens in about 5% of spinal taps. But one thing that we want to emphasize is that that's about the same as when we looked at data from the Red Cross for a blood donation. So, kind of the nausea and the dizziness would be the same as a blood donation. So I know Dr. Fagan suggests that we should call this a spinal fluid donation. So just touch a little bit about what factors contribute to these complications when we see them. So this is one instance where definitely being older is better. So the older that you are, there's actually fewer complications from lumbar puncture. So the greatest risk of a lumbar puncture headache is in a very young person. And we found, interestingly, that if you had had a lumbar puncture headache once, that actually did not increase your risk for having a second lumbar puncture headache. Now, some people do get two, but statistically, there was actually no increase in risk if you've had one headache. So I just want to give some take-home messages here. So I think the thing that I've been most excited about from this survey was that previous experience with lumbar puncture increases the chances that people will agree to a second one. So as I said, I think that just emphasizes that we just need to educate people, let people know that it's not, that you're not going to be paralyzed, that it really doesn't hurt if everything goes the way that it should. So we just need to get that message across and not have it be such a scary event to people. That lumbar puncture headache is relatively rare in older adults, and usually the headache is just of mild severity. And that as far as the risk of any other complications, it's almost identical as far as getting a whole blood donation. So I want to, again, as Dr. Moore said, to encourage you to come back and agree when you get called for follow-up and even baseline if you haven't considered it before. And I'm sure there's lots of folks here who have, both from the Memory and Aging Project and other participants who've had lumbar puncture and have had a good experience. So thank you. I apologize, I got my ear a little bit off. So we just want to get a quote. This has just happened, I think, just a couple weeks ago. We had someone call and leave a very nice voicemail. A gentleman called and said, I am in Dr. Morse's Memory and Aging program. This morning I had a lumbar puncture done by Dr. Nupur Ghoshal, who I know is here somewhere also. Dr. Ghoshal gets super credit, with assistance from Sushi Sathian, who I haven't seen this morning. I just wanted to tell you that they made the whole procedure almost pleasant, if you can say that. They are terrific in what they do. They made me feel very comfortable. You should be very proud of them. So we have an amazing team. All of our lumbar puncture physicians are neurologists, and certainly Sushi is wonderful, and Dr. Fagan and the whole team. So it's almost pleasant. And just as far as the survey that we did, I wanted to thank all of the sites, all the Alzheimer's centers that contributed, all the coordinators, the clinicians, and all the participants that helped us learn more about your attitudes towards lumbar puncture. Definitely the Alzheimer's Association to give the funding, and again, the Knight ADRC team who helped with this. So most of you know Sushi, who's helped organize your lumbar puncture, and Becky Fearberg. So thank you.
Thank you, Krista. By the way, we've been doing this participants breakfast for a long time and trying to figure out the best way that a group of your size in a room this large can have good access to viewing the information and the screens. And we have just a terrific group, Ron, Holly, and Howard Palmer, who get all this set up and functioning so well. So Ron and Howard are in the back, and let's thank them. So John Cerrito is a Knight ADRC investigator and has been part of a study to determine whether certain antidepressant medications may have benefit in Alzheimer's disease. So Dr. Cerrito is going to present this. Let's welcome Dr. Cerrito.
Thank you. Thanks everyone for coming this morning, surviving the little rain we had. So I want to talk about some studies we've been doing both in mice and in people. My lab studies, probably most of what we do is studying Alzheimer's disease in mice, trying to understand this amyloid beta protein, why it's made and why it's cleared from the brain. And I'll tell you a little about those studies today, understanding how antidepressants, which a lot of people are on, might be useful, could be useful, we'll see, in this whole fight against Alzheimer's disease. So my lab wants to understand how neuronal activity, our brain functioning normally, regulates how much A beta gets made. And from a variety of research we've been doing over the last 7 or 8 years, we found that neurotransmitters, these chemicals that are responsible for the electrical signals that occur in our brain that we need for thinking, might control this protein. And serotonin is one of these neurotransmitters that we thought was important. So antidepressants or selective serotonin reuptake inhibitors, SSRIs, increase the serotonin chemical in the brain. So we had the question, very, very easy question to start. If we gave mice, Alzheimer's mouse models, an antidepressant, what would happen to this A beta protein? And when I was a graduate student with Dave Holtzman, we developed a technique to measure A beta in the brain of mice over time. This is so you can measure every hour over the course of days. So we measured normal A beta levels in a mouse over the first, 8 hours or so. And then we gave just a single dose of an SSRI. Now we've got 5 different SSRIs, and in all 5 cases, what happens is, if you look at A beta levels every hour, A beta levels go down over a 24-hour period. Actually, by 12 hours, they're decreased, and they stay down for at least 24 hours. So that supported our hypothesis that perhaps in these SSRIs, by increasing serotonin, you could lower this A beta protein. This happens for these are Alzheimer's mice. So mice don't normally get Alzheimer's disease. They have the A beta protein. We give it, but mice don't generally get plaques. So we give them the human protein. So at young ages, what these experiments I'm showing you here are, these mice don't have plaques yet, but when they get 8 or 9 months of age, they'll start getting plaques. We wanted to then know what happens so is this selective for serotonin? That's what we originally thought. There are antidepressants that don't act through serotonin. In this case, we gave drugs called reboxetine or teneptine. These are non-SSRI antidepressants, and these don't change A beta levels. So if we did the exact same experiment I just showed you, but at time 0 we gave these antidepressants, and over 24 hours there's no change. So it's not that these mice are happier. It's not a mood effect that lowers A beta levels. It really is an effect of this, we think, this chemical serotonin that was now specific to this. In studies that I'm not going to tell you about, I had a graduate student that did a lot of the cellular signaling, what's going on actually inside the cell. We understand a lot of the molecular biology, the very small, minute things that are going on in the cell that make this happen. So we have a lot of understanding about why serotonin is actually changing A beta levels. And in mice, it's great to be able to show that these things happen. We can do a lot of experiments we can't do in people. But ultimately, we want to know what people care about. What's going to affect Alzheimer's disease is not just A beta levels in young mice or in young people, but what's going on in old people or old mice that have plaques. So we had a technician in my laboratory that took these Alzheimer's mice that before they had any kind of plaques in their brain, gave them an SSRI, or in this case citalopram, in their drinking water for 4 months. And what we found is if you take, they were either in drinking water, regular drinking water, or the SSRI. And at the end of when these mice were 8.5 months of age, we took the brains out and then stained for this amyloid beta protein in plaques. And if you look at the picture on the left is regular mice. And a normal mouse would have none of this brown staining at all. All these brown dots are A beta plaques throughout the cortex and the hippocampus, the areas of the brain that are involved in memory as well as cognitive function, both in people and in mice. And even by the pictures you can tell, in the mice treated with citalopram, there's a lot less brown in the cortex and hippocampus than we had otherwise. And then what's on the bottom are the graphs of this, so you can actually then quantify how much plaque these animals had. We also took the cerebrospinal fluid from these mice. This is the lower right-hand corner. And very similar to what you would do in a person, we did in a mouse. And the amyloid beta levels in the mouse also went down when you had a very long treatment of citalopram. So this is consistent, and if we can lower A beta levels for a short period of time in our mice, if you give it long periods, we actually can affect plaques. We thought that was very promising. But again, curing a mouse of things is actually reasonably simple in a lot of cases. Doing this in a person is really the critical thing we want to understand. So as we were doing our mouse studies, completely unrelated to this, Yvette Shaleen, who used to be in psychiatry at Washington, now she's at UPenn in Philadelphia, came to my office out of the blue and said, we're doing a study looking at amyloid imaging in depressed patients versus non-depressed patients. Are you interested in doing parallel studies in your mice? I was like, well, we're already doing that. What are you doing? And she said she had a study that was going on for a couple of years at this point, where she had taken, and she was a psychiatrist who had been seeing depressed patients. She had taken those people and did PET imaging to look at their amyloid beta levels using this Pittsburgh compound B, or PIB. And the original hypothesis, which ultimately proved out to be wrong, was that people that were depressed would have more plaques in the brain for a lot of different reasons. They have a lot of factors that increase stress, among other things that might increase plaques. And she found the exact opposite. The people that were depressed actually had less plaques in their brain than people who were not depressed. Then when she went back and she was comparing that to people from the ADRC, people that are not depressed but had PET scans done. And she went back and found that anybody who had any treatment with antidepressant in the last 5 years actually had less plaques in their brain. So it wasn't tracking whether they were depressed or not. It was tracking if they had any history of use of an SSRI. And here's actually imaging, an image of a PET scan. Normally a PET scan would have very little yellow or red. In this case, you can see in people that were depressed, they have lots of red and yellow in their brain. That's the imaging, this PIB imaging that we're finding. But this is in a lot of cases a retrospective set of data that we went back into our archives, the data that people have already had these scans done. We already know is you do a prospective trial that's randomized, that you control for, and ask people that if you give them antidepressants, can you change your A-beta levels or not? So we did a technique that was developed here by Randy Bateman several years ago, where you can label newly synthesized A-beta in cerebrospinal fluid. So we did this. We brought people into the hospital late at night, and we had them in the hospital for 36 hours. We gave them a single dose of an antidepressant, in this case again citalopram, the same drug we were giving our mice, and then measured cerebrospinal fluid A-beta levels every hour for 36 hours. And we found that if you look at the open circles on the top are people that were treated with placebo, and the bottom are A-beta levels for people that were treated with citalopram. You can see that over the entire course of our measurements, A-beta levels were significantly lower in people treated with the SSRI. This is essentially the mouse experiment I showed you on that first slide, now done in a person, measuring A-beta every hour over time. So again, very promising that we can use an SSRI to lower A-beta levels. What we didn't do from this study, which is unfortunate now looking back at it, is that we actually started sampling at time 8 after we gave the drug. What would have been great if we actually sampled at time 0. We actually saw that A-beta level decrease over those first 8 hours. So we need to go back, and we actually are now going back and looking at those first 8 hours. But the data is promising, at least, that we can affect A-beta levels in a person very similar to what we're seeing in mice. These people were all cognitively normal, young individuals, and between 20s and early 50s. If you're going to treat someone for Alzheimer's disease, you're not going to treat this population. College kids, you're not going to really treat for AD at this point. So the study that we have going on now is in older individuals, people that are between 65 and 80, that are still cognitively normal, don't have symptoms of Alzheimer's disease. A lot of these people very likely have A-beta plaques that are in their brain already. Asking whether we can see a change in cerebrospinal fluid A-beta levels in that population, people that would matter, people that we would need to treat. This is a very slightly different study design. It's not measuring A-beta every hour over days. It's bringing people in, taking a sample of their cerebrospinal fluid, and then treating them for 2 weeks with the escitalopram. It's actually a derivative of the citalopram drug. And at the end of the 2 weeks, bringing them back in and taking their cerebrospinal fluid again, and asking over this 2-week period, can you see a change or not? So that study is currently being enrolled both here at WashU as well as at UPenn. Joyce Snyder's helping out the WashU site. So that study is ongoing. We don't have results from it yet, but we're actually actively recruiting, and I think our recruitment is going very well for that study at this point. So I want to conclude by saying a few things. One, and we think that we understand how these drugs are affecting A-beta levels. It's not just some random phenomenon. We know that the chemical in the brain is playing a role, serotonin. We know how that chemical is doing this. That in mice, if we give an SSRI, you can lower A-beta levels, you can lower plaques, which is promising, but treating a mouse for things is actually, like I said, I won't say simple. It's definitely not simple, but certainly simpler than doing it in a person. In that SSRIs in people seem to be doing the very similar things that we're finding in our mice. But most people don't frankly care whether they have amyloid plaques in the brain or not. They care if they have symptoms of Alzheimer's disease or not, whether they have memory or not. So the real study we haven't done yet, the study where we give people SSRIs probably before they start showing symptoms, and see can we prevent or delay symptoms, that study still has to be done. I think we're very hopeful that the studies we've done so far will lead us to that. But we're not at a point where we're ready to start putting people in SSRIs to treat them for Alzheimer's disease. Again, we can treat a mouse for things, we can cure a mouse of lots of things, including for Alzheimer's disease in many cases. And a lot of those things so far haven't panned out for Alzheimer's disease in people. But we are gearing up to start doing that study, hopefully do that study. So at some point, maybe SSRIs could be used for this purpose. So with that, I have a laboratory that does all these studies. very rarely get to touch a mouse these days. And then of course all of you, and you guys have taken part in this study as well, so thank you very much.
Well, thanks so much, John. Some of the brand names for these drugs, citalopram is Celexa, escitalopram is Lexapro. Another one of the class of this class of drugs is Prozac. Any of you heard of some of those names? Recognize? All right, so let's move ahead. We're going to talk about something that we haven't presented here ever before, about circadian rhythms. And Eric Music is assistant professor in the Department of Neurology and did a postdoctoral fellowship with us in Dr. Holtzman's laboratory, and is going to talk about what circadian rhythms are and why they may be relevant for Alzheimer's disease. So, Dr. Music.
Good morning. It's great to see so many people here. So let me start by kind of introducing the idea of circadian rhythms, and I'm going to tell you what I'm going to tell you in the first slide. So what I'm going to try to explain is what circadian rhythms are, and how they regulate sleep and are critical for health in general. And I'm going to tell you a little bit about how they're disrupted in Alzheimer's disease, and that may lead to some of the sleep abnormalities we see in the disease. And then I'm going to tell you about how the body creates these circadian rhythms through the function of genes in our DNA called clock genes, and what happens in mice when you disrupt this system. We don't know in people yet, but in mice, certainly if you disrupt the system, you see damage in the brain, which is reminiscent of that seen in Alzheimer's disease. And then I'm going to talk a little bit about at the end about how we hope to target this system to perhaps stave off some of these effects that we see in Alzheimer's disease. So you may have heard of circadian rhythms. Circadian rhythms have come around because we all live on Earth, or at least most of us live here on Earth. And Earth has a 24-hour light-day cycle. So almost every animal that lives above ground has adapted to this 24-hour cycle by developing circadian rhythms. And these rhythms, circadian it means about a day. So it means a one-day rhythm, a 24-hour cycle in our body. And our body has these rhythms to try to synchronize what's going on inside our body and inside our cells with the external environment, and to synchronize what's going on in one organ with another organ. So everything's happening at the right time of day in our bodies. So there's a part of our brain called the suprachiasmatic nucleus, which you don't need to remember, but that's the master clock of the brain. And so the eye actually senses light, and it tells the suprachiasmatic nucleus if it's light or if it's dark. And the suprachiasmatic nucleus is like the main clock of the brain. It sets these rhythms at the normal 24-hour cycle. So if you were to get on a plane after this meeting and fly to Europe, you'd notice that the light-dark cycle shifted by several hours. And so for a few days, your brain would be thinking it's 5 o'clock, and the light-dark cycle would be telling it something different, and you'd feel a little bit out of sorts. So we call that jet lag, where your body is not in synchrony with what's going on in the environment. But the beauty of the circadian clock is after about a week, your brain would catch up, it would resynchronize to the new light-dark cycle, and everything would be okay again. So it turns out that almost every cell in our body has its own internal circadian clock. And so the cells in your liver have a clock. So certain things happen in your liver at night and during the day that are different. And these cycles are all kind of synchronized together by the suprachiasmatic nucleus. So I like to think of it like an orchestra. And each organ is like a person playing an instrument. They can all play their instrument independently, but the suprachiasmatic nucleus is like the conductor of the orchestra that keeps everything synchronized. So it turns out that within each cell in our body, almost every cell in our body, we have a cellular clock that keeps this 24-hour time. And there's a picture of it here. You don't need to memorize all the names of the genes. But the point is there are specific genes in our DNA that actually dictate our circadian rhythm. And if you look at a cell, actually you can take a cell out of your body and put it in a plate, in a dish someplace where there's no light or dark cycles, and it will still continue to show circadian rhythms. It'll show rhythms in how it functions. It'll show rhythms in what genes are being expressed different times of day. So these circadian rhythms are really fundamental to how our cells work. So why am I telling you all this? This is not a talk on basic biology. But it turns out that circadian rhythms are important in aging, and we think they're important in Alzheimer's disease. So it's been known for a long time that as we get older, our circadian rhythms are less robust. You don't sleep quite as well at night, and you're more sleepy during the day, sleep being one of the circadian rhythms. So it turns out that it's not just the sleep-wake cycle that's a circadian rhythm, but it's many other aspects of our bodily function. So our hormone secretion, our body temperature, our heart rate, blood pressure, all these things are regulated by the circadian clock. So we often think of sleep and the circadian clock as being the same, and certainly they're intimately connected, but they're not exactly the same. The circadian clock actually regulates sleep and lots of other things. So if you look at some of these parameters, one of the easiest things to do in people is that you put a little wristwatch on people, kind of like a Fitbit or an Apple Watch or one of these other things that now you can buy over the counter. And it can sense how much a person moves in a day. This is called actigraphy. And so this is a picture of what you get when you look at these watches later. You can see all these little dark spots. That means that this person is very active during the day, and then there's this light area, meaning that they're sleeping at night. And each one of these lines going across, I realize why this is so hard to use, it's left on the left side. Each one of these is a 2-day cycle. So you can see this person day after day after day is active during the day and they're sleeping at night. There's these really nice columns. They have really nice circadian rhythms. They're very well defined when they're sleeping and when they're awake, when they're active, when they're not. This is a person that doesn't have Alzheimer's disease. This at the bottom is a person that does have Alzheimer's disease. So you can see the person with Alzheimer's disease, if you look at those two tracings, they don't have those nice columns anymore. Everything is a little more disorganized. The person is more awake during the night, getting up many times, and during the day they're less active, sleeping more. And this normal night-day cycle is broken down. So we've known about that for a long time. And in fact, many of you who have been around people with Alzheimer's disease have probably experienced this. Your loved one may get up in the night, think it's the middle of the day, they may sleep a lot during the day, and you've probably seen this with your own eyes. But what's interesting is a new study came out recently that suggested that if you look at people that don't have Alzheimer's disease, just older people, and you do these same kind of analyses and you look at their circadian rhythms, that older, they particularly looked in women in this study, then they looked at thousands of women, they found that older women that had poorer circadian rhythms or less organized circadian rhythms were more likely to develop dementia in the next 5 years. So it suggested that this problem may actually precede the disease and is something that we should look into a little bit further. So the main reason why I'm here right now is because David Holtzman, who you've already heard about several times, had done some really pioneering work on sleep and Alzheimer's disease. And in collaboration with Randy Bateman, the two of them had shown that in mice and in humans, that amyloid beta protein, this plaque-forming protein that we've heard about today, shows these day-night fluctuations. And so this is a picture, that's not going to work. A picture here of a plaque, and this is the amyloid protein going up and down and up and down and up and down in the brain of a mouse. And Dr. Bateman showed the same thing in spinal fluid from humans, probably from some of you in this audience, suggesting that the sleep-wake cycle actually controls the levels of this amyloid protein in the brain. And Dr. Holtzman went on to show that if you sleep-deprive mice, you don't let them sleep normally, you disrupt this process, that they get more plaques, and if you give them drugs that increase sleep, they get fewer plaques. So his lab had shown this really beautiful connection between sleep and Alzheimer's disease, or at least sleep and plaques. And so we wondered, well, does the circadian clock play a role in this process? And we're still wondering that. This research is still in process. What we do know about the circadian clock is that it's important for health in other parts of your body. So there are large studies of people who have poor circadian function. So these are people who work the night shift, or people who are flight attendants on international flights and are constantly jet lagged, people who don't have a normal circadian rhythm. These people are at higher risk for diabetes, for coronary artery disease or heart disease, for cancer, for a variety of other diseases. So it turns out it's not good for you to have disrupted circadian rhythm. You want to kind of keep that rhythm strong. But we don't really know what happens in the brain of these people. There's a few studies suggesting that people with poor circadian rhythms have worse memory, but no one had really looked very carefully at this. So I won't show you all the data, but some of our initial studies, we used our knowledge of the genetics of the circadian clock and made mice that were genetically engineered to have no circadian rhythms. So these mice, they actually are not sleep deprived. They sleep the same amount as a normal mouse, but they just sleep the same amount every hour. They don't have a day or a night. It's just they catnap all day. Strange things to say for a mouse to catnap. They do. So when we knocked out the circadian clock genes and made these mice totally arrhythmic, we found that they got inflammation in their brain, that some of the nerve cells were injured and the synapses were damaged, that they have learning and memory problems, and that they have increased free radical damage in the brain. And that these are many things that we see in Alzheimer's disease as well, certainly not good things in the brain. So we've hypothesized that by disrupting the circadian clock, that may actually worsen some of the effects of amyloid beta in the brain and may actually cause inflammation and nerve cell damage to become worse. So one of the main reason why we're trying to understand this connection is to see if we can do something about it. Can we fix the broken clock? Can we target the circadian system with some kind of behavioral intervention or a drug or something that will help revitalize these circadian rhythms and perhaps protect the brain against this injury? And so we have some leads. This data on this, really not critical to understand this, but a recently published paper showing a new drug that's been developed. It's still an experimental drug that actually targets the clock directly and can sort of rejuvenate these rhythms. And as luck would have it, the person who developed this drug moved from California to St. Louis in the past year, and so we're collaborating with him to try this in mice with the hope that someday we'll be able to target the circadian system in people to perhaps prevent the disease. So the take-home points of what I've been saying. We think that circadian rhythms and certainly the sleep-wake cycle are important for protecting the brain from Alzheimer's disease, and that disturbing the circadian rhythm may lead to sleep problems and other problems that may predispose the brain. It may set the stage for Alzheimer's disease. And we're using mice, and we now have some funding from the National Institutes of Health to look in people, in yourselves, and in data that we've already collected actually from people's spinal fluid, and some of you out there may have worn some of these little wristwatches, but there's a bunch of data that's been collected where we can see if in humans, if the connection between disrupted circadian rhythms and increased Alzheimer's disease biomarkers holds up. And then finally, we hope to ultimately try to test drugs that target circadian clock to try to prevent Alzheimer's disease. So there's a lot of people that work on this with me. I have to thank Dr. Holtzman and Dr. Morris, who are my two mentors here at WashU, and I have to thank all of you for participating.
Thanks very much, Eric. So I mentioned at the outset that we're interested in prevention of Alzheimer's disease, and we're always looking for experimental therapies to see if they can help us in our fight against Alzheimer's. And Dr. Joy Snyder is here to talk about an update on what we're doing with these experimental therapies. Dr. Snyder?
Good morning, everybody.
Good morning.
So I'm going to talk about people because, unlike Dr. Cerrito and Dr. Music, I'm not unhappy that I don't deal with mice anymore. So I'm very, very glad that I'm a 100% people doctor now. So I'm going to talk a little bit about clinical trials, but I wanted to start off by laying the groundwork for that, and again reminding you, which I probably don't have to, but how important it is that we do take on Alzheimer's disease and how great it is that you are playing such a major role in doing that. This is a slide that I got from Dr. Morris, and basically what it shows is that we've made a lot of progress in healthcare in the U.S. and the world. It's been fantastic. We've reduced deaths due to breast cancer, prostate cancer, heart disease, stroke, lots of things, but not Alzheimer's disease. As the population ages, more people get Alzheimer's. Deaths from Alzheimer's is going up, not down. So this is critically important work. It's awesome that you are doing it for us. It's awesome that your tax dollars come to Dr. Morris and the rest of us so that we can do this work, but there's a lot of work left to do. And to emphasize the diversity point, it's also important that we do study Alzheimer's disease in everybody, and particularly in African Americans, they are overall more likely to get Alzheimer's disease. So we have to do this work, we have to do it in everybody, and we can't do it without you. So you're the most important thing. And as Dr. Music said, that's really my bottom line. Some of the studies I'm going to tell you about are not actually ones you can be in, but they're ones that you made possible through the work you've done over here over the last 30 plus years through our Alzheimer's Center. So it's a great thing. So to reiterate some of the things that Dr. Morris said, we've undergone a change in the last few years in Alzheimer's disease, and as usual, with the many years I've been working with Dr. Morris, he's kind of leading the charge in changing how the field thinks about Alzheimer's. And we now think about Alzheimer's not as just the part of the disease where people have memory problems, because that's just the tip of the iceberg. The disease starts 20 or 30 years before, and we're trying to use the term Alzheimer's disease to refer to that whole time period when the brain changes start years before symptoms. That's really important because that may be how we're going to finally make big moves in treating this disease. So Alzheimer's disease is the whole spectrum from when those brain changes start all the way through to when people do ultimately develop symptoms. And it has two stages. It's got that preclinical stage where there are no symptoms, you don't know you have a problem, but in your brain the plaques and tangles are starting to develop, and then there's the symptomatic stage. And until the first prevention studies were started 3 years ago, every clinical trial focused on that symptomatic stage when there had already been changes in the brain for 20 or 30 years. A lot of those trials didn't work, as you know, probably because the things we were trying to target were things that had already happened. So we're having to rethink our approach there and thinking more about preclinical treatment of those plaques and tangles and maybe other targets later on. The other key thing that our center, I think, has taken the lead in, and the reason that many of you here are study participants and the other half are study partners, is that it's really important for us to understand how a person changes. When you come and get your psychometric testing that you hate more than the lumbar puncture, I'm happy to hear, but when you get that testing, you don't really care how you compare to the average person. You want to know how you compare to you, what's changed about you. So we focus here on intra-individual change, how each person has changed from the way they were. Some of you come in and complain about having trouble with remembering people's names. That sounds really weird to me because I've never been able to remember anybody's name. But for some of you, that's a big deal, and I appreciate that. So we need to hear from your loved ones, people who know you well, what's changed about you. So we focus on intra-individual change, how you have changed. That also makes it really important, as you heard, to do follow-up, because we can't tell how you've changed on all those measures unless we compare you to you. So again, that's where the follow-up becomes really important. We compare you to how you did last year, 3 years ago, 5 years ago. So that's really important. So this is, you've seen this before. I think Dr. Mulder and Dr. Morris both showed copies of this, but this is the Alzheimer's disease timeline. And what we've learned, and where's that big red dot, is that one of the first changes is in that spinal fluid amyloid beta peptide. And really, this is, you guys can all pat yourselves on the back, especially if you've had a spinal tap. We've learned this in large part because of you. As Dr. Mulder said, we have the biggest collection of spinal fluid in the world. We have the most longitudinal data probably in the world. So we've learned this because of you. This is kind of fuzzy though, right? This is -15 to -25. That's a pretty big range. We still need you to help us nail that down. That's why we need those longitudinal studies. So 10 years from now, when somebody's at risk for Alzheimer's and we do a spinal tap and we get their numbers, we can say your risk in 5 years is 42%. Give them some number they can listen to instead of saying, well, maybe 10 or 20 years. So right now we don't have that defined yet, but you can certainly help us do that. We also have taken the lead in amyloid imaging, in measuring tau, both in spinal fluid and in imaging, and a lot of things you didn't hear about today, those MRIs that everybody also says many people don't like those because you're in the tube, but we get beautiful pictures of your brain. We can do careful measurements, see how much certain parts of the brain shrink. So all those things change later, and then here at the very end is when we start seeing changes in your memory and thinking, which we measure using this clinical dementia rating sum of boxes. So this is just a picture of the amyloid imaging. You've seen, I think Dr. Cerrito showed some of these, but in a normal person, we don't see a lot of bright stuff. They don't have amyloid in the brain. This is measured using Pittsburgh compound B, but we can also use this new compound called 4-beta-pir or amyvid. In a person with Alzheimer's, you see a lot of that red stuff. That's those amyloid plaques. This is that autopsy without having to be dead that Dr. Morris was talking about, where we can see amyloid in the brain. And here's a normal person, but they look just like the person with Alzheimer's disease. And what's interesting, and again, this is data from our center, this is data that you and the folks who were in the study before you have helped us do, we now know that this normal person got Alzheimer's 3 years later. So this helps us predict, using the imaging, who's going to get Alzheimer's. This is using, again, I mentioned 4-beta-pir. It's called amyvid. It's now commercially available. When I see a patient in the clinic who's worried about Alzheimer's, I could order this study. I could order it, but their insurance wouldn't pay for it right now, so it probably wouldn't happen. But that's something that new studies are going to go forward and see if this is going to be useful. But it was approved by the FDA. It is available. And if we see a positive scan, it means they have amyloid plaques and probably have Alzheimer's disease. We can see this, again, both in people with symptoms and without. We don't quite yet know how to interpret it in people without symptoms in the clinic, but we can use it for clinical trials, and that's what I'm going to tell you about next. So the A4 study, you guys have heard about the A4 study. Whenever I tell people about it, I have to struggle to remember what all the A's are. Anti-amyloid against Alzheimer's, I think is right.
No.
No? Oh, it's
Anti-amyloid asymptomatic.
see, I can't even read my own slide. Anti-amyloid asymptomatic Alzheimer's, just because I don't have my cheaters on. And the key point of this study is to figure out, in people who are at risk for Alzheimer's, who have that positive amyloid scan, can we treat them with drugs that target amyloid and keep them from getting Alzheimer's? So this is a prevention study. We're trying to prevent Alzheimer's, which would be a really cool thing to be able to do. It's using an anti-amyloid antibody. It's called solanezumab. And we're treating people in this study for 3 years to see if they can not get Alzheimer's disease. And they're also having all those other studies that you all love. They're having optional spinal tap. They're having amyloid imaging, all that great memory and thinking testing that everybody hates. So lots of things going on in this study. There's going to be 550 people worldwide, or I guess nationwide. The study's being led by Dr. Sperling out of Harvard, and we're a site here, and Dr. Bateman is our site PI. It's got to be a lot of people, right? Because these are people without symptoms, and very few of them will develop symptoms. So over 1,000 people have to be in the study. Half of them will get the drug, half will get placebo. Study doctors don't know who gets which. It's an IV drug, so everybody comes in once a month and gets an IV. So here's some sort of key facts about that study. People are going to be 65 to 85 years old. They have to be normal, no significant thinking problems. Have to have a study partner, same as for our study. We want to know how inter-individual change is going. And they have to be willing to do all that crazy stuff. Have the MRI, cognitive testing, amyloid imaging, and consider having the LP. Going to come in 3.5 years, once a month, so pretty frequent visits. So Dr. Morris said we're asking a lot. This study is asking even more than we usually ask. The study is focused on diversity, getting people who are at high risk, getting a diverse population, getting people over 75, people with a family history, lots of features. So how is this going to work? So we're going to take people who are cognitively normal, and in order to get the drug, they have to have that positive amyloid scan. So guess what? We have to screen a lot of people to get one person for the study. So it's very intensive, and Erin Couture is back there waving her hand. She's standing up, but she's not very tall. So
keep waiting.
She's waiting. Erin is the study coordinator, and she has already been working incredibly hard, screening lots of people for this study to get folks enrolled. So it's ongoing. It's a very exciting study. It's a study to prevent Alzheimer's, the first one in people without a gene mutation, but it is tough. So you can absolutely contact us about it. There's a website. Get in touch with Erin. Let us know if you're interested. We're going through and recruiting people. Don't everybody expect to come see us next week, because Erin might run away. But we are getting through this slowly. It's a great study, and we're very excited about it. We are a little worried about it, as Dr. Morris said, from the other aspects, because once you're in this study, you kind of move out of the memory and aging core study. So we're a little concerned about that. We're also recruiting people who aren't in this study. If you have friends in the community who are interested, who are over 65, they can contact us. We're recruiting patients from our clinic, so we're going everywhere to try to recruit folks for this study. The biggest reason to tell you about it is because you, in large part, made it possible. Because of the studies you did here at Memory and Aging, the imaging, the spinal taps, all that stuff you put up with, we laid the groundwork to make this study possible. So you were the biggest single cohort in the world who made this study possible, and you deserve our thanks for that, and hopefully you're part of what will eventually prevent Alzheimer's disease. So other studies. So what about studies in people that already have symptoms of Alzheimer's disease? We're not giving up on them. It's been a long, hard road. Lots of studies have come and gone, but we still want to treat people who have the symptoms. The one study that we are participating in now, and you got to love these study acronyms. They must have a committee somewhere that comes up with this stuff. But this is the SNF study, Soluble Nasal Insulin to Fight Forgetfulness. And it gets even better because the study drug is something you squirt up your nose. So this is the SNF study. And the idea here is that there's some crossover between Alzheimer's disease and diabetes, and a lot of information has been going on about that the last few years. But it turns out in Alzheimer's disease, the brain may not regulate insulin properly. So insulin may play a role. And insulin, besides being very important for your pancreas and glucose metabolism, is also a growth factor for the brain. So in this study, people who don't have diabetes, you can't have diabetes to be in the study, people who don't have diabetes and do have Alzheimer's will squirt insulin up their nose and see if that helps the brain. So it's a way to deliver insulin pretty much directly to the brain. It's a 12-month study where people are randomized, and then another 6 months where everybody gets drugged. The problem with this study is it turns out it's kind of hard to reliably squirt insulin up your nose. So right now the study is on hold while they're trying to find the correct device to do that. But we do hope to be recruiting for that study as soon as they get the device sorted out. And I think Joyce Haney is the coordinator for that study, and she's waving over here, and she will be around later if anybody wants to know more about that. And there's a lot of other studies on people with Alzheimer's going on. If you go to clinicaltrials.gov, there's 200 plus. We can't do them all here. As Dr. Morris said, we have a limited pool, and we're very protective of you. You may not think so based on all the stuff we do, but we really are. And so we can only do a few studies, but there's certainly a lot going on. I don't want you to think there's not any action in Alzheimer's. There's a lot going on, and we hope to have better treatment soon.
Okay, go quickly for this last one.
Okay. So very quickly, a couple of other things. Dr. Morris mentioned the Dominantly Inherited Alzheimer's Network and the Dominantly Inherited Alzheimer's Network Treatment Trial. It's led by Dr. Randy Bateman worldwide. It's a fascinating study. You're not going to be eligible for it because these mutations that cause the early onset Alzheimer's, if you guys saw Still Alice, that was what Still Alice had. Those mutations are very rare, but we were able to launch that first prevention study here a couple of years ago, and that's very exciting. And again, you made that possible. The groundwork we laid in your studies and your spinal fluid, your imaging, made this study possible as well. So it's a very exciting study, and we hope to continue trying to treat that disorder. You heard from Dr. Cerrito about the Shalem study. This is doing the study in people that was done in mice, studying a drug called escitalopram. And I think one of my participants from that study this past week is here. She had an LP and then had another one 2 weeks later and lived to tell about it. So it's a very exciting study. We're recruiting 30 people here at WashU, and these are folks who get treated with an antidepressant for 2 weeks with an LP at the beginning and end to see if the things Dr. Cerrito was talking about in mice actually work in people. And again, it's for people without memory problems. And then in the future, so the future is we hope it's bright. You know, I hate to try to be overly optimistic and say we're going to cure Alzheimer's in X amount of time, because I've learned over the last few years we really can't say that. But I think we are making a lot of progress. And the emphasis is on these biomarkers, the imaging and the spinal fluid, and that's where you've played such an amazing role. And the emphasis is on getting people who are normal and trying to prevent Alzheimer's. So we're very hopeful that this is going to be a promising time for us. And it's all made possible because of you. So The final thing is there are more studies coming down the pipe. The A5 study, I think I heard Dr. Sperling mention plans for A6, so I don't know who's going to come up with all those A's, but we'll see what happens. Other antibodies are coming down the pipe, so lots of exciting things are happening. We're very excited about it. We hope to participate in it, and we want to, ultimately, I think that's my last slide. One more. Bottom line, yes. So, as I already said, it's very exciting. People with mild impairment, people that are pre-symptomatic, preventing Alzheimer's. So imagine, as the Alzheimer's Association says, a world without Alzheimer's. That would be great. We don't want to live forever, but we want to live well, and I think that would be a way for many people to do that. We hope to continue doing that, and we can only do that with your continued help. So thank you so much. I look forward to seeing you all. I may have a sharp object in my hand when I see you if you come for your LP, but we'll still have a good time. Hopefully it will be even pleasant, not just almost pleasant. So again, thank you very much.